Oh boy, we got some exciting stuff happening here. Chapter 8 is all about photosynthesis. And this thing, my heart always is moved by this chapter because there's this great symbiosis that happens between us and plants, and we help keep each other alive. And without one or the other, all life as you know it would cease to exist. And so this is kind of a cool topic. I really enjoy talking about it. So the first first an overview of photosynthesis. Here's a little bit, of how it works. Sunlight's going to hit a pigment that is in leaves, and what happens is that water is taken from the ground, and also kind of the air, the vapor, the water vapor in the air, and carbon dioxide is taken from the atmosphere. And so you basically are taking water, you're adding a little carbon dioxide, you're adding a little bit of sunlight, and then the big product is that sugars are made, and also as a byproduct, oxygen is made. So that's kind of the long and the short of it. Here's the equation. You take 6 molecules of carbon dioxide, add to 6 molecules of water. Okay, so here's the carbon dioxide, here's the water. You add a little bit of sunlight, that's this, and you end up with some sugar, and then 6 molecules of oxygen as a byproduct. Now you're probably going to remember or probably recognize this formula. You're going to think to yourself, oh, I've seen that somewhere. That's because photosynthesis equation is nearly the exact opposite of the cellular respiration. So the aerobic respiration, I guess, is a form of aerobic. Respiration equation looks something like this. You take 6 molecules of, oops, already off on the wrong foot. You take glucose, you add to it 6 molecules of oxygen. That is going to be broken down and makes, oops, sorry. I'm really blowing it here. Here we go. 6 molecules of CO2 plus 6 molecules of water plus ATP. So you have glucose adding to oxygen and make carbon dioxide, water, and energy, so to speak. But in this equation, you have carbon dioxide being added to water, and you add a little bit of energy into the mix, and you make sugar and oxygen. So you can see it's just kind of a flip-flop. And what's really great about that is that you have this beautiful symbiosis that's happening. Symbiosis means a relationship between living things which is mutually beneficial, meaning it's good for the one organism and also good for the other organism. So how cool is this? That when we make energy, we make carbon dioxide and water and energy. Well, guess what it is that plants need? They need water and carbon dioxide. And we are more than happy to give them these byproducts that we're making. Now, keep in mind the water that we actually make in our cells is not truthfully the water that plants require, because plants will take water from like rivers and streams and things like that, whereas the water that we make in cellular respiration is going to mostly stay in our cell. But still, the CO2 that we produce, all of the CO2 produced by living organisms does go to kind of contribute to the plants. So plants really like CO2. And they are going to make sugar and oxygen, which we definitely need these things. All right, so really, in essence, every glucose molecule you ever consume probably originally came from a plant. And also, all the byproducts that plants make is going to provide the oxygen that is vital to our well-being and our existence. So it's this really cool relationship where all of the byproducts are utilized and all of the products are utilized. And this is part of the reason why I like spending a lot of time in the woods. If you're like me, you're just going to sit next to a tree and just think about how great those trees are and how great all the plants are, because they're keeping you alive. But I digress. Let's get into it here. So here's what's going on. There is a leaf, and just kind of want to orient you where this whole photosynthesis process is occurring. It's occurring generally in the leaves of the plant, as opposed to like the stems or the branches or the trunk. And if I was to take if I was to look at, here's a cross section of the leaf, and I was kind of to look at a small chunk of the side of it, it would look something like this. So this would be like the top of the leaf, and this would be kind of like the bottom of the leaf. All right. And if you look at kind of the microanatomy of a leaf, there's a layer of cells on the top and there's a layer of cells on the bottom, and in the middle there's just a bunch of plant cells. What's kind of interesting though is in the bottom what's called the lower epidermis. I'm not really too concerned that you know all of these names of the anatomy of a plant. We'll save that for plant biology. But in the bottom of the plant, there's these little holes called stomata, and this is going to allow the gas molecules to enter and exit. So CO2 comes in, and oxygen comes out. And if you wanted to see what an actual stomata looks like, here's a scanning electron micrograph, so picture taken with an electron microscope of the underside of a leaf. And you might see one of these little tiny, teeny little microscopic holes in the bottom of those leaves. You're not going to be able to see these with your eye, but they do nevertheless exist. So this oxygen or carbon dioxide gets into the leaf, and then carbon dioxide gets into the leaf, and oxygen leaves the leaf. Well, if you were to take a look at just one of the plant cells and magnify it, it would look something like this. You know, looks just like an animal cell for most part. It's got a nucleus, it's got ribosomes and mitochondria. And it does have this unique organelle called the chloroplast. This is a plant's only organelle, or at least it's not really found in animals. You might make the exception for a couple of animal-like protists, but in the animal kingdom at large, there is no chloroplast found in any of the cells. So if I was to look at the chloroplast, which is an organelle, and magnify it, it would look something like this. There's a couple of features I want to point out. First is that they are almost a little bit similar to a mitochondrion in the sense that they have an outer membrane and an inner membrane, and that was true of mitochondria. Inside of the chloroplast, then, are these little discs called thylakoids. And if you stack a whole bunch of discs up, it looks kind of like a big stack of pancakes known as a granum. And this is kind of where photosynthesis is occurring. There's a lot of it occurring kind of along the thylakoid membranes, and then there's a lot of it occurring in this empty space here too, which is filled with this fluid called, which we call the stroma. So that's where it's all occurring, photosynthesis. Now, what's happening in photosynthesis, there's two parts to it. There is what's called the light-dependent reaction and the light-independent reaction. Light-dependent means that it requires light. So this is what's happening during the day when the sun is out and shining on the leaves. But plants can also do a whole bunch of stuff during the darkness, but even during the light, they can still do this type of reaction, but it just doesn't require light. It can happen with or without light. Another thing that this is named is the Calvin cycle. Okay? So light-dependent reaction, light-independent reaction, which is also called the Calvin cycle. Here's a real brief overview of what's going on in these two reactions. So what you see here, this is a big enlarged chloroplast. So this is an organelle that's inside of a cell, a plant cell. And over here we have what's showing the light-dependent reaction. And over here this is then the light-independent reaction. And the main thing that's going on with these is that in the light-dependent reaction, light is being used. And the main result is that it's going to make ATP and it's going to make this stuff. Now you're going to see this molecule and you're going to go, oh, that looks really familiar to me. And that's because when we talked about aerobic respiration, we talked about this molecule called NADH. And NADH was an electron shuttle, right? It just was something that carries electrons around. Well, the electron shuttle that we use in photosynthesis, or that is used in photosynthesis, is not NADH but NADPH. I don't know exactly what this stands for. I mean, a simple Google would tell you it, but off the top of my head I don't. But I like to just remember that the P is what makes it unique, and so I usually just recall that the P stands for photosynthesis. Now it's not actually what it stands for, I don't think, but it does help to remember this. So in either case, you're making some energy and you're making some electrons. That's what you make in the light-dependent reaction, energy and electrons. Well, the fate of the energy and the electrons is that they get dumped into this thing, which is called the Calvin cycle, named after the scientist Calvin, who probably discovered it. And the Calvin cycle is the thing that's going to produce this stuff, which stands for glyceraldehyde 3-phosphate. In other words, when you see this, you just think to yourself, sugar. The molecule that we call glucose is made from two of these what's called glyceraldehyde 3-phosphate. But we'll talk more about that later. Okay. So in summary, you take some energy and some electrons, which are made in the light reaction. It's brought to the Calvin cycle to make glucose. And then what happens is that you have an empty electron shuttle and some byproducts here that need to be converted back into ATP and electrons. So that's kind of this dance that's happening. You send the NADP plus and the ADP and PI. These get formed into ATP. This gets formed into NADPH, which is what you see here, and then the whole cycle just happens and continues and continues and continues. The main benefit is that as this whole dance is occurring, we're producing sugar. Okay, so this the light independent reaction and the light independent reaction, there are a little bit more to discuss about them, but if you ever get confused about what it is we're talking about when we talk about light independent or light dependent, this is kind of a good picture to reference. Just remember, ATP and electrons are made in the light dependent reaction, and sugar is made in the light independent reaction. So the first thing we'll talk about is the light dependent reaction. This is the most probably complicated of them. So forewarning, we're gonna have quite a bit to kind of slug through as we talk through the light dependent reaction. The first thing we should talk about is light. You know, if it requires light, we got to get our heads on straight about what light is. And light, that's kind of tricky because nobody really knows what light is. Some people think it's a wave, some people think it's a particle. The best, because there's different experiments that show either way. But the best kind of model that we use to figure out what's going on with light is we think of generally these particles called photons. And photons kind of move in these waves like this. And they just travel like this throughout space. Okay, so right now there's these photons that are coming off of the sun, and they are traveling in these little waves millions and millions and millions and millions of miles to finally land on our beautiful green Earth. Okay, now because they travel like this, they form what are called waves, and the distance between these peaks is called the wavelength. And there's a variety of wavelengths out there that make up what's called the electromagnetic spectrum. Okay, so this is called electromagnetic radiation or electromagnetic waves. Here's a little picture of what's known as the electromagnetic spectrum. So for example, if the distance between these two waves of the photon oscillating or moving back and forth, if it's on the order of kilometers or meters, then we're talking about radio waves. So some radio waves are really huge. You know, there's a photon that moves 10,000 kilometers before it makes its way back up to the top of the wave. So the space between the waves is like 10 kilometers or thousands of kilometers, okay? Or it could even be on the order of meters. Well, once you start getting less than a millimeter, so on the scale of nanometers, there's a range that we call visible light. So the difference between violet and blue and green and yellow and orange and red and all of the colors that you see out in the world is really just where these photons are, the wavelength between the crests of the movement of the photons. So some receptors in our eyes detect some wavelengths, and then we'll perceive it as something maybe like violet and blue, whereas other receptors in our eyes can detect other types of wavelengths, and they'll see it as perhaps yellow or orange or red or green or any of these colors. So they're all different wavelengths. here's kind of an example. So like red has the longest wavelengths, so the length between the photons that are moving up and down is maybe the longest, and then violet type light is going to have, the shortest wavelength. And that's not, you don't really have to know what has the longest wavelength and the shortest wavelength. The big idea that I'm just trying to show you is that there are different types of light that have, or rather, there are one kind of light that has different wavelengths, and the variety of wavelengths out there are going to create the appearance of the different colors that we see. And this is important because plants have certain molecules which can absorb only electromagnetic radiation of certain wavelengths. So that gets us to these molecules called pigments. So a pigment is any molecule which absorbs visible light. And depending on the molecule, some of them are going to absorb a certain wavelength and others are going to absorb a different wavelength. So here's a picture showing a bunch of different pigments that you might find in plants. These are the big ones, chlorophyll A and chlorophyll B. Chlorophyll is kind of the big champion here. But there's also some other ones. There's like carotenoids and phycoerythrin and phycocyanin. And this shows kind of their absorbance throughout the different wavelengths out there. So what you're seeing on this graph is this is how much percentage of the light is absorbed by the pigment. And so if you take a look, chlorophyll B is a big one that's in plants, is going to absorb pretty much anything between this 450 to 460 range. So you see it kind of right up here. About 80% of the light is absorbed if it's between these wavelengths. And so that's kind of like bluish light. And you can also see that it absorbs, over here if I come across, about 20% of the light is absorbed that's within kind of the reddish colors. So if you have blue and red colors absorbed, then the ones that are reflected are the ones that we see. So this is kind of an interesting thing. Again, this isn't something that you have to know for the exam, but something that you might just care to know about because it's interesting. Leaves, the reason why they're green is because these pigments, chlorophyll A and chlorophyll B, all right, so here's chlorophyll A right here, you can see that their peak absorptions are kind of in this wavelength and in this wavelength. So the things that they reflect are green and yellow. And together, that's why when we look at plants, they have a green color because they're not really reflecting any of the blues or any of the reds. It's mostly just the green. Well, what happens in the fall, the reason why the colors turn as they do in the fall and have these, nice vibrant yellow and orange colors is because chlorophyll A and chlorophyll B, these ones are the first ones to die off when the weather gets cold. So then you're left with the carotenoids. And then also there's another type of pigment that's actually produced in the fall, and the carotenoids and the one that's produced in the fall, they are going to reflect the reds and the yellows mostly, and they are not going to. There's not going to be any of these chlorophylls reflecting green light, so that's why the colors change as they do. All right, so pigments are molecules that absorb light. If you have a whole bunch of pigments in a small region of space, we call it a photosystem. So photosystems are basically they're just like these proteins or regions where there are lots of pigments associating close to one another. There's a high concentration of the pigment molecule. Here's an example of what a photosystem might look like that's inside the thylakoid membrane of a chloroplast. Basically, there's a whole bunch of little pigment molecules, and what happens is that light will strike onto these pigments, these pigment molecules. And specifically, the main thing that happens in a photosystem is that as light is striking all of these pigments, they're all getting excited. And once they reach a certain level of excitement, there's two that are in the middle of this photosystem, and when all of these are kind of donating their excitement to the two pigment molecules in the middle, eventually these get so excited that they pop out an electron. Okay, so the electron goes boom and it comes out. Well, in reality, it lands on this thing called an electron scepter, but we'll talk. We won't talk about that right now. But the key idea is that light hits these photosystems and an electron pops out. And then, well, here's kind of an interesting that happens. In order to replenish the electron that's been popped out, electrons are put into the photosystem. This happens through a process known as photolysis or photolysis. In photolysis, if you look at the word, it literally means light breaks. Photo means light, and lysis means break. And photolysis, the light breaking light, what it does is it actually is going to break apart the molecule water. So water gets broken apart. Its electrons are what are used to replenish the electrons that are lost out of the photosystem. Now, there's a couple of different photosystems in photosynthesis. There's photosystem 2 and there's photosystem 1. The one that I've just described here is photosystem 2. But there's another one, photosystem 1, which looks like this, and they have different jobs. They behave very similarly, but they just have a very kind of subtle difference in their main job, and I will explain those differences to you in just a minute. But I do like to point out, photosystem 2 is usually the thing that happens first in the process of the light-dependent reaction. Photosystem 1 is the thing that happens second. So I apologize, this is super confusing. You know, photosystem 2, you want to think that 2 is the thing that comes second, but in fact photosystem 2 is the thing that comes first, and then photosystem 1 is the thing that comes second. So these things were named not in the order in which they kind of occur in this whole long process, but rather in the order that they were discovered. All right, so none of this probably makes a whole lot of sense up to this point, which that's fine, that's okay. I'm going to now show you the whole process, and then hopefully you'll understand what's going on with these photosystems. So here we go. Here's what's going on. What we have here is a thylakoid. Okay, so inside the thylakoid, we call it the thylakoid lumen. Outside of the thylakoid is the stroma, that's that fluid, and then this is all happening inside of a chloroplast. So here is photosystem 2, right here. And what's happening is light is striking the pigment molecules, the chlorophyll. Now when that happens, they all get excited, and then these ones in the middle pop off their electron, and then the electron is going to whiz across the electron transport chain. So here you see it. You're going to see this and you're going to go, ah, I know what that is. Or hopefully you say, ah, I know what that is, because this was something that was found in cellular respiration as well. Okay. And the electron transport chain in photosynthesis has a very similar job as it did in aerobic respiration. The electrons, as they come across the electron transport chain, they're going to pull in the hydrogens, and they're going to push them against their concentration gradient. If you take a look, you can see that in this figure you're seeing a high concentration of protons inside the thylakoid, and a lower concentration of hydrogens or protons outside of the thylakoid. So the energy that's required to push something up its concentration gradient is these negatively charged electrons flowing through the electron transport chain. So this is a very similar story to aerobic respiration. Okay, so as those are coming across, these electrons are luring in the hydrogens, and then they're kind of tricked into this space where there's a lot of hydrogen ions. Then what happens is the electrons are going to make their way to photosystem 1, which is this one that you see right here. What happens at photosystem 1 is that light is going to strike the pigment molecules, and again it's going to excite the two pigments in the middle, and then they're going to pop out the electrons, which, so the electrons came in, and then they get excited, and they pop back out again, and then they make their way to this combination of molecules, which this is kind of what you might think of as the empty electron shuttle. And then this molecule is what you might think of as the full electron shuttle. All right. The empty electron shuttle, it picks up some electrons and then it's off. And then the whole point of this is there's two reasons. One, you want to make these. You want to gather up some electrons because they're going to be used for something in a minute. And then the other purpose of all of this is that you're accumulating all of these hydrogen ions in here. And the same story as aerobic respiration, the hydrogen ions, they want to flow from high concentration to low concentration. And in this scenario, there's a high concentration of ions in the middle of the thylakoid, and there's a low concentration of ions all around the outside. So they actually flow from inside of the thylakoid to the outside. And in so doing, they're flowing through an ATP synthase, which is exactly the same story that we had in cellular respiration. The flow of protons through that ATP synthase is going to take an ADP, it's going to take a P, it's going to link them together to form ATP. All right, so you see that two things are really made in this whole process. You have NADPH made, and you have ATP made. Those are really the kind of the two things. And in the process, water has been split, to keep supplying electrons to this whole process. And in that, oxygen is formed. And you might see that this is kind of the exact flip-flop of what happens in cellular respiration. In cellular respiration, oxygen is the molecule that's taking up the electrons coming off of the electron transport chain, and when that happens, it actually forms water. But that's not the story here in photosynthesis. In photosynthesis, water is releasing electrons and making oxygen, and then also some hydrogen ions. So a pretty cool process in that it's kind of just the total reverse of what's going on in aerobic respiration. So now here's my question to you. We've made some ATP, we've made some ADPH. Now the question is, where on earth did these things go and what purpose do they serve? Okay, this is a big, what's going on here? Where do these go? And if you recall, if we go back up to this picture that I showed you just a little while ago, right here, you see here the two things are. This is what we make in the light-dependent reaction. We make ATP and we make NADPH. This is energy. This is electrons. And for whatever reason, there's this cycle called the Kelvin cycle, or the light-independent reaction, that needs to take ATP and needs to take electrons to make this, which is sugar. So if we go back down here and answer our mysterious question, the answer is where these electrons need to go and where this energy needs to go. The answer then is that they have to go to the Kelvin cycle. And their purpose is that they are going to make some good old-fashioned, they're going to be at least involved in making sugar, or specifically glucose. Okay, so that's kind of the story of the light-dependent reaction. So we've got all sorts of fun stuff here to unpack. I kind of threw a lot at you, so we're going to kind of unpack some of these things. Here are some equations, or here are some questions that I'm asking. What is the equation for photosynthesis? Draw a chloroplast, label a few things like the inner membrane, the outer membrane, the thylakoid membrane, the lumen, and the stroma. What is the function of a stromata, not to be confused with a stroma, which is a very similarly sounding name. What is a pigment? And then describe the light-dependent reaction of photosynthesis. Where does it occur? What is going on? What are the reactants? What are the products? How does it work? This is a big one, so you're going to have to spend a little bit of time with that. Go ahead and pause the video and work on these, and then I'll tell you the answers. All right, the answers are as follows. The equation for photosynthesis looks something like this. You take 6 carbon dioxide, they are added to 6 water, and a little bit of energy is required in the form of sunlight. And this is going to produce some glucose. And then as a byproduct, a little bit of oxygen. So 6 molecules of oxygen. Draw chloroplast. Chloroplast looks something like this. There's an outer membrane. and this membrane would be a phospholipid bilayer. Then there's another membrane, an inner membrane. There are these little discs called thylakoids. And there's a bunch of fluid in here that's not part of the discs, and that is called the stroma. That's basically all of the rest of the space here, the fluid. And then the lumen would be the very inside of the thylakoid. And then also, if we have a whole stack of these, we call it a granum for whatever reason. I'm not exactly sure why it's called that. Okay, a stomata is not the same as the stroma, nearly identical looking word, but it's a totally different function. The stromata is the small hole that you see in the underline of the, on the underside of the leaf. And this is going to allow the entrance of CO2 and also the exit of oxygen. CO2 enters, O2 exits. What is a pigment? A pigment is a molecule that is able to absorb light. And there's a bunch of different types of pigments, and they do the type of light that they absorb, whether it be blue or yellow or red or orange or green. It all depends on the type of pigment. The big one that you generally hear of is chlorophyll. And chlorophyll is going to absorb mostly blue and then also some red light, so that means that it reflects green and yellowish light. And then lastly, describe the light dependent reaction of photosynthesis. Here is the big one. All right, this is the big one. And let's take a quick look at this. So it's helpful to think about the things that are going into this portion of photosynthesis and the things that are being made. So we're talking about the light dependent. So it's definitely going to require some light, right? It depends on light. And some of the other things that are involved is that water is involved. You have these little components, ADP plus PI. Keep in mind, this isn't going to be a balanced equation, so it's not showing the entire, all of the atoms being balanced and everything, but it does kind of help you just keep straight what's going on. Also included is NADP plus H plus. All right, and then some stuff is going to be made. The ADP and the P, they get put together, so they make ATP. Okay, so this kind of gets converted into this stuff. These, they receive a few electrons, and they get made into DPH. And then also, what's going on here is that water breaks down through a process known as photolysis. In the process releases some oxygen. And also probably I should include some hydrogen ions as well. All right. So here's what's going on then with this whole thing. There's a photosystem 1, or sorry, photosystem 2. There's a whole bunch of little protein things that together form what's called the electron transport chain. There's another photosystem called photosystem 1, and then there's a couple more little protein things which make up an electron transport chain. And then there's also a ATP synthase. These are kind of the big players. And this is all found along the thylakoid membrane of a thylakoid. So those are those little disc-like structures. So I'm drawing kind of, this is the phospholipid bilayer here that I'm drawing right now. So this would be the lumen of the thylakoid. And then all of this space outside of the thylakoid would be called the stroma. And then all of this stuff is inside of a chloroplast. And there's a whole, keep in mind, there's a whole bunch of these thylakoids inside one chloroplast. All right, so here's what happens. Light is going to strike the photosystem 2. That's going to excite a lot of the pigment molecules. You know, there's a whole bunch of pigment molecules in here, and those can absorb the photons in this light. They get really energized, and then out pops 2 electrons. They go, I should write it like this. You got these electrons that go poof, and they pop out of here, and they race through all of these proteins in the electron transport chain, and then they make their way to photosystem 1. And then light strikes photosystem 1, and all of the pigments that are in photosystem 1 get all excited, and these 2 electrons that have just arrived, they get excited, and then they get pushed out of photosystem 1, and then they are going to continue along through this little miniature electron transport chain, and then they're going to kind of jump off onto the electron shuttle. So incoming is NADP plus H plus, which is kind of we always kind of write these together. This is the empty electron shuttle, and it's going to come by, it's going to pick up those electrons, and it's going to form NADPH, the full electron shuttle. So that's where we get this coming from. Where we get oxygen coming from is that in order to replenish all of these electrons, which are continually being excited and passing along, you have water, which needs to be split into hydrogen ions and also oxygen, and that's going to be what releases these electrons into the photosystem. This is called photolysis or photolysis. So where you see this oxygen, that's why we have this as a byproduct, and that's also why we have these hydrogen ions as a byproduct. The whole reason of all of this is because the electrons are passing through, and in that pass, they are attracting hydrogen ions to go get pumped up their electron, or up their concentration gradient. So inside the lumen, there are a whole bunch of hydrogen ions, and these get pumped into there from low concentration to high concentration. Then there's one last thing that needs to happen in this whole dance of biochemical activity, and the answer is that the hydrogen ions need to now flow down their concentration gradient through the ATP synthase, and when they do that, they take ADP, they add a little phosphate onto it, and it makes ATP. This is the ATP synthase is the machine that kind of takes these two and puts them together to manufacture ATP. The fuel for this machine, the thing that makes it run, is all of these hydrogens flowing through it. So it's really a pretty cool, fascinating thing that's going on here. So basically, there's a few things that you have to keep in mind. You would want, if I was to ask you, imagine on an exam I said, describe the light-dependent reaction to me. There'd be a couple or quite a few things I'd want you to describe. I'd want you to say, well, light hits photosystem 2, it excites electrons, they pass through the electron transport chain. When that happens, they pump hydrogen ions up their concentration gradient into the lumen. The electrons then are transferred from photosystem 1 to reduce NADP plus H plus to make NADPH. And then lastly, that the hydrogen ions would then flow down their concentration gradient out again to make the ATP synthase work in order to make ATP. I know that sounds like a lot, and it certainly sounds like a lot too, because a lot of these molecules are kind of a mouthful to say. But in the end, the story is actually pretty simple, believe it or not. Electrons pass through, that establishes a concentration gradient that makes ATP, and in the process, some electrons are made that need to go somewhere. That's really the whole story, if you take out a lot of the nuance. All right, so that's the light-dependent reaction. So we're saying again, we are making ATP, we're making NADPH, we're making some oxygen, we're making some hydrogen ions. These things here, ATP and NADH, can you remember where these go? We said that they go somewhere, but what is the purpose of all of this? The purpose is to send some electrons and some energy to the Calvin cycle, and that's because the Calvin cycle is the place where we will make some sugar. So let's go ahead and talk about it. The Calvin cycle. This is the light independent reaction. It can happen during the night, or it can happen also during the day. It doesn't matter. It doesn't require light to be present. So here's how it works. We said that electrons are made and energy is made from the light dependent reactions. These get brought over to the Calvin cycle. The actual Calvin cycle looks something like this. There's 3 phases to it: fixation, reduction, and regeneration. And there's kind of a lot of things that are listed here, but I'll walk you through it, and I hope you find it's not too complex. It's not too hard to kind of get your mind around. The fixation, reduction, and regeneration, 3 phases. So what happens in fixation is a 1-carbon molecule gets attached to a 5-carbon molecule to make a 6-carbon molecule. Okay, if you take 1, you add 5 to it, it makes 6. Now the names of these molecules: CO2 is the 1-carbon molecule, and this molecule called ribulose bisphosphate, or RUBP, is the 5-carbon molecule. So here's where this is happening. You have CO2 from the atmosphere that's diffused into the leaf of the cell, and ribulose bisphosphate. This is a 1-carbon molecule. This is a 5-carbon molecule. You take a 1-carbon molecule, you add it to a 5-carbon molecule, you make a 6-carbon molecule. Okay, this is the first step. So the way that plants build sugars is they build them 1 carbon at a time. They continually run this cycle round and round and round and round and round, and every time it runs, 1 carbon is being added into the system. And this is just this continual construction of these sugar molecules through carbon dioxide. So how cool is that? That plants can just take this gas that's all over the place in the atmosphere, and just 1 carbon at a time, 1 CO2 molecule at a time, piece it by piece to make a sugar molecule. I think that's pretty cool. All right, so then the 6-carbon molecule, it has kind of an interesting fate. It undergoes reduction. And my first question here for you is, what does that mean? Remember, reduction is the opposite of oxidation. So reduction is a gain in electrons. Remember, if you can't remember what the difference between reduction and oxidation is, remember that Leo the lion gurs, meaning a loss in electrons is oxidation, whereas a gain in electrons is reduction. Just kind of a fun little mnemonic there to help you remember the difference between them. So if we're talking about reducing something, we're talking about it taking up some electrons, all right? So here's what happens in this scenario. You have this 6-carbon molecule which immediately gets broken down into 2 3-carbon molecules called G3P, which stands for glyceraldehyde 3-phosphate. Okay, so this is a 3-carbon molecule and this is a 3-carbon molecule. You've got 2 of them there. Now to make the 6-carbon molecule get broken down into 2 3-carbon molecules, you have to pour a bunch of stuff into the cycle. So here's where we get our products getting used up. The products from the light-dependent reaction, this is where they end up. They come down here. ATP is going to release its energy to drive this whole thing to happen. And NADPH is going to take its electrons over to here and dump its electrons into the system. So if you're putting in energy and you're putting in electrons, you're constantly putting in energy and electrons, you can make this 6 carbon be reduced and then split into the 2 3 carbon molecules. Now, why this is important is because we end up making some of this stuff. This 3 carbon molecule here, glyceraldehyde 3-phosphate, when you start accumulating these, you can actually add 2 of these molecules together to produce glucose. Okay. Now the one that's left behind, we said the 6 carbon molecule forms into 3 carbon molecules. The one that's left behind gets actually regenerated back to our original BUPB, which is this whole process here, the regeneration phase. Okay. So the last review question of this chapter is list and describe the 3 main steps of the Calvin cycle. Okay, so first thing you want to be able to do is list them. So here they are, 1, 2, 3. See if you can just take a moment to guess or just figure out, hopefully you're not guessing, see if you can do this on your own, list the 3 main steps of the Calvin cycle. Alright, so here are the answers. The first one is fixation. The second one is reduction. The third one is regeneration. And here's kind of a pretty simplified version of the Calvin cycle, but one that's still helpful. In the fixation phase, you're taking a 5 carbon molecule, known as ribulose bisphosphate. You're adding a 1 carbon molecule, CO2, from the atmosphere, and you're combining these together to form a 6 carbon molecule. This is the first step, which we call fixation. The next thing is that the 6 carbon molecule is going to get split into 2 3 carbon molecules. And the name of these 3 carbon molecules is glyceraldehyde 3-phosphate, or G3P, we call it. And to make this whole process happen, I guess I should say, to make this whole process, we need to put a couple things in. We have to put a little bit of energy into the system. We also have to put a few electrons into the system. This is in the form of ATP coming and breaking off its energy and releasing it, and then forming ADP plus P. And then the electrons, this is in the form of NADPH coming off, letting its electrons loose, and then NADP plus H leaving separately. But it's just kind of a fancy way of saying we're putting energy and electrons into there. Since we're adding electrons, we're gaining electrons, we can say that this is reduction. So this whole step here is going to be step number 2. And then the last step is that this 3 carbon molecule, one of the 3, gets converted back to RUBP. Now you might think, wait a minute, how do I get a 3 carbon molecule just magically turning back into a 5 carbon molecule? And the answer is a little bit more complex, and this is kind of where the Calvin cycle gets a little bit more tricky, and it's a level of trickiness that I don't really want to worry about in this class. But if you're wondering exactly how it works, one turn of this will create 2 G3Ps, but it's not really as simple as I've drawn here. Really how it works is that it takes 3 of these turns of the cycle to produce 1 of these externally. So oftentimes this will turn once, and then the 2 of these things will actually be combined to then return the ribulose bisphosphate. But that's neither here nor there. It's a little bit more complex. But if it goes 1, 2, 3, and one of these pops out. And then it goes 1, 2, 3, and another one pops out. And it takes 2 G3Ps, take G3P, add it to a G3P, and you make this stuff called glucose 6-phosphate, which is basically glucose. Right? It's a form of glucose which can be linked up together to form starch. But glucose is made. And And so here's kind of if you're doing 1, 2, 3 turns to make one of these molecules, and then you go 1, 2, 3 turns to make another of the molecule, then in total it takes 6 turns of this Calvin cycle to make one molecule of glucose. So there we have it. That is the Calvin cycle. I've got a really great animation here that I'll leave you with that kind of shows the whole story.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of 2 sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life.